History of Life on Earth. Chapter 5, Tissues, Organs, and the Cambrian Explosion. The world fills up. 5.1 The Fold. Here is the single most important event in your personal history, and it happened before you were 3 weeks old. You were a hollow ball. Not a promising object, a few hundred cells arranged into a sphere with nothing inside it. A soap bubble made of biology, indistinguishable to the naked eye from the earliest embryo of a fish or a fly or a sea urchin. And then, on one side of that ball, the cells did something. They moved. A patch of the surface buckled, dimpled, and began to fold inward, pushing in and down like a thumb pressed into a balloon, until the invagination met the far wall, and the hollow ball became a tube within a tube. That fold is called gastrulation, and the biologist Louis Wolpert made a famous remark about it that has never been improved on. It is not birth, marriage, or death, but gastrulation, which is truly the most important time in your life. He was not being cute. Before the fold, you were a mass of cells. After it, you had an inside and an outside, and crucially, you had layers. Sheets of cells with fates. The cells on the outside would become skin and nerves and brain. The cells swept inward would become the lining of your gut. And the third population, appearing between them, would become nearly everything else you can point to. Muscle, bone, blood, heart. Every animal above the grade of a sponge does some version of this. The fold is the moment a mass of cooperating cells stops being a colony and starts being a body. Chapter 4, Got Life to the Sponge. Cells that stick together, specialize, and die for the collective good. This chapter takes the fold and the layers it makes and follows them until the world is full of animals. It ends with an event so abrupt that the first geologist to see it in the rocks assumed something was wrong with the rocks. 5.2 What a tissue actually is. A sponge has cell types. It does not have tissues, and the difference is worth being precise about because it's the whole subject of this chapter. A tissue is not just a bunch of cells doing the same job. 3 things separate a tissue from a crowd. Its cells share an address. They don't decide what to become individually, ad hoc, wherever they happen to end up. They come from a defined region of the embryo, a germ layer, and their fate is determined by where they came from. They are arranged, not scattered. Organized into sheets and bundles and layers, held in position, often glued down to a specialized mat of extracellular matrix called a basement membrane. Architecture, not soup. They do something together that no single cell could do. A sheet of gut lining absorbs nutrients from one side and passes them to the other, a directional act that requires the sheet to have sides. One cell cannot have a direction. A tissue can. Recall from Chapter 4 that you can push a sponge through a mesh and let it reassemble itself. That is not a party trick. It's a diagnosis. A sponge's cells can be scrambled and rebuilt because they were never really arranged in the first place. Try that with a jellyfish and you get jelly. That is the line we are crossing here. Past this point, structure becomes irreversible, and irreversibility is the price of complexity. 5.32 Layers. The jellyfish plan. The simplest surviving version of the fold produces 2 layers. Biologists call it diploblasty, and it is the body plan of the cnidarians: jellyfish, corals, sea anemones, hydras. The outer layer, the ectoderm, becomes the skin, and remember this, it will matter enormously, the nervous system. Nerve and skin are siblings, born from the same sheet, and they will stay strangely entangled for the rest of this book. Your brain is, developmentally speaking, a piece of skin that folded inward and never came back out. The inner layer, the endoderm, becomes the lining of the gut. And between them, filling the space, is the mesoglia, a thick, transparent jelly, mostly water and protein, with very few cells in it. It is not a tissue. It's packing material and it's structural. It's what gives a jellyfish its bulk and its bell and its springy resilience. It is why a jellyfish is 95% water and still holds a shape. Look at what this plan can do. A sea anemone is not a simple animal in the dismissive sense. It has real epithelium, organized sheets with a top and a bottom. It has real neurons and real synapses. It has stinging cells so beautiful and terrible that they deserve their own paragraph. The cnidocyte is arguably the most sophisticated single cell any animal has ever built. Inside it sits a capsule under enormous osmotic pressure up to 150 atmospheres, comparable to the inside of a fire hose, containing a coiled inside-out harpoon. When the trigger is touched, the capsule fires. The harpoon everts, accelerating at something on the order of 5 million grams, and punches through the cuticle of a crustacean or the skin of a fish in under a millionth of a second. It is among the fastest movements known in the entire living world, and it is executed by a single cell, once, using stored pressure, a cell that spends its whole existence loaded like a gun and then, having fired, dies. That is what 2 germ layers can do, but look at what they cannot do. Cnidarians are radially symmetric. They have a top and a bottom but no front, no back, no left, no right. They have one opening, which serves as mouth and anus both, so that food goes in and waste comes back out the same door. They cannot burrow. They cannot chase. Their nervous system, as we'll see, has no center. Corals are anchored to rock. Anemones barely move. Jellyfish drift, pulsing, mostly at the mercy of the current. They are 2/3 of an animal. What's missing is a layer. 5.4 the middle layer. Nearly every animal you have ever noticed, every worm, insect, snail, crab, starfish, fish, frog, bird, and mammal, belongs to a single enormous lineage called the Bilateria. And what defines it? Beyond having a left side that matches a right side is a 3rd germ layer, wedged in between the outer and inner ones during gastrulation. The Mesoderm. This is the great innovation. Ask what Mesoderm builds and you will see immediately why this chapter treats it as the hinge of animal evolution. Muscle, nearly all of it. Blood and the vessels that carry it. And the heart. Bone and cartilage, every skeleton in the vertebrate lineage. Kidneys. Gonads. Connective tissue, the tendons, the ligaments, the fascia, the structural bulk of a body. Put a hand on your chest. Everything under the skin, apart from the lining of your airways, and gut in the nerves threading through it. All of it is mesoderm. The thing you push against when you push against yourself. 2 germ layers give you a bag with the nervous system. 3 give you an animal, something that can push, grip, chase, burrow, flee, and bleed. And once you have mesoderm, an organ can become a real organ. Not a fold of lining, but a genuine machine with its own musculature, its own blood supply, and connective tissue holding it in shape. You cannot make a heart out of 2 germ layers. You can barely make a muscle. When did this happen? Deep in the Precambrian, before the fossils are any use to us. The first Bilaterians were almost certainly tiny, soft, and utterly unpreservable, millimeter-scale things crawling in the mud. They left us essentially nothing. But when they finally surface into the fossil record, they surface everywhere, and all at once, and we'll get there. 5.5 room inside. Having a middle layer raises a question that turns out to matter enormously. What do you do with the space? 3 answers exist among the Bilaterians, and each one buys a different kind of animal. Fill it in. Flatworms are a columette, solid, packed with mesodermal tissue from gut to skin. No cavity at all. This works, but it constrains you brutally. Everything must diffuse through solid tissue to reach every cell, and diffusion over distance is hopeless. So the animal must stay thin. This is why a flatworm is flat. It isn't a style choice, it's a respiratory requirement. It has no room for organs to grow, no space for a circulatory system, nowhere to put anything. Leave a gap. Roundworms are pseudo-columate. There's a fluid-filled cavity in there, but it isn't fully lined with mesoderm. It's a compromise, better than solid, but the organs float free in it, unsuspended, and the muscles of the body wall and the gut can't act independently. Line it properly. And then there is the cellum. A true body cavity, fully enclosed by mesoderm on both sides, mesoderm against the body wall, mesoderm against the gut, with the organs slung between them on delicate sheets of tissue called mesenteries. This is one of those innovations that sounds like a technicality and is in fact a revolution, because a properly lined cavity gives you 4 things at once. A hydrostatic skeleton. Fluid can't be compressed. Squeeze it in one place and it must bulge somewhere else. An earthworm burrowing through packed soil is exploiting this. It squeezes at its circular muscles and goes long and thin, drives forward, then squeezes its longitudinal muscles, and goes short and fat, anchoring itself. Long, thin, push, short, fat, grip. It is boring through the earth using nothing but muscle and incompressible water, and it can generate real force doing it. Room to grow. Organs can enlarge, fold, and elaborate into the empty space without deforming the animal's outer shape. Your gut is roughly 7 meters long. It fits inside you because there is a cavity for it to coil in. Independence. With the gut suspended in fluid rather than mortared into the body wall, your intestines can churn while you sit still, and you can run while they keep digesting. Locomotion and digestion are decoupled. 2 systems, 2 jobs, running simultaneously. Somewhere to put a circulatory system. Once the body has an interior space, blood can be piped through it, and a body with a blood supply is a body that can be thick. It can be large. It can build bulk muscle and feed it. There are, incidentally, 2 different ways to construct a coelom. The mesoderm can split open from a solid block, or the cavity can bud directly off the primitive gut, and which route an animal takes turns out to divide the whole bilaterian world in 2. That split is Chapter 6's opening move, so hold on to it. 5.64 Fabrics. Out of these 3 layers, in every bilaterian on Earth, cells specialize into 4 basic kinds of tissue. Everything in your body is one of these 4 things. Epithelium is the tissue of surfaces, sheets of tightly bound cells that cover, line, protect, absorb, and secrete. Your skin. The lining of your gut, your lungs, your bladder, your blood vessels. Epithelium is the odd one out because unlike the others, it isn't defined by where it came from. It can arise from any of the 3 germ layers, depending on where in the body it's needed. What defines it as polarity? An epithelial cell has a top and a bottom, and they are not interchangeable. The top faces the world, or the gut cavity, or the airway. The bottom is anchored to the basement membrane. The cells are sealed to each other at their tops so that nothing can leak between them. Think about what that buys you. A sheet with a top and a bottom is a barrier and a pump. It can grab a molecule on one face and release it on the other. It can decide what gets in. Every boundary in your body, between you and the world, between your blood and your gut, between your air and your blood, is an epithelium doing directional work. And the whole thing is built out of the Cadherin adhesion system from Chapter 4, just organized with far greater precision. Connective tissue is the tissue of the spaces between. Sparse cells scattered in a great deal of secreted matrix. It's the exception to everything you know about tissues because it's mostly not cells. Bone, cartilage, tendon, fat, and, surprisingly, blood are all connective tissues. Blood is connective tissue in which the matrix happens to be liquid. Muscle is the tissue of force. It contracts and nothing else in the body can. Nervous tissue is the tissue of information. Cells that signal at speed and integrate what they receive. The last 2 are so consequential for everything that follows that they each get a section. 5.7 The invention of the nerve. Sponges have no neurons. They have cells that can signal slowly and locally, a whisper of what's coming, but nothing that fires. Cnidarians have neurons. Real ones. And this is one of the great mysteries of the animal tree, because the neuron does not appear to be a small step. It arrives more or less complete. A neuron is a cell that has taken the ordinary electrical properties of a membrane. Every cell has a voltage across it. Every cell pumps ions, and weaponizes them into a signal that travels. It builds a wave of voltage that propagates along its length without weakening, and at the end of the line it converts that electrical signal into a chemical one. It dumps a puff of molecules across a microscopic gap, the synapse, onto the next cell. The receiving cell reads the puff and decides whether to fire in turn. Electrical, then chemical, then electrical again. That is the language of every thought you have ever had, and it was fully operational in something like a jellyfish before there were eyes to see it happen. In cnidarians, the neurons are arranged in a nerve net, a diffuse mesh spread evenly through the body, with no brain, no center, no headquarters. Poke an anemone anywhere and the signal propagates outward through the mesh in all directions and the whole animal responds. This sounds primitive, and in an important sense it isn't, it's appropriate. A radially symmetric animal has no front. It doesn't face anything. Threats and food can come from any direction with equal probability, so there is no reason to concentrate your processing at one end. A nerve net is the correct answer to the question a jellyfish's body is asking. And to make sure you don't file this away as simple, consider the box jellyfish. It is a cnidarian. It has a nerve net. It also has 24 eyes, arranged in clusters around its bell, and 4 of them are true camera eyes, with a cornea, a cellular lens, a retina, and an iris that contracts in bright light. They form images. Box jellyfish have been observed navigating deliberately between mangrove roots, using their eyes to see the canopy overhead through the water surface. They hunt. They steer. They have no brain. Sit with that for a moment. It's a good corrective against the tidy story where complexity marches upward in a straight line. Life is far weirder and more resourceful than that. But the fully centralized nervous system, the one with the head, with the sensors bunched at the front a swelling of nervous tissue right behind them to process what they report, has to wait. That's a Belatarian invention, and it requires something a jellyfish does not have: a front end. Once an animal habitually moves in one direction, the end that goes first meets the future first, and it pays enormously to put your eyes there and your brain right behind them. Brains are what happens when a body starts going somewhere. 5.8 The invention of muscle. The molecular machinery of contraction is very old, older than animals, older than nerves. Actin and myosin were introduced back in Chapter 3, in the cytoskeleton of a single cell, where myosin walks along actin filaments hauling cargo, and where actin's assembly and disassembly pushes and pulls the cell into shape. Muscle is that machinery scaled up and organized. Cnidarians take the first step with myoepithelial cells, epithelial cells that keep contractile fibers at their base. They're still part of the skin sheet, they haven't left it, but they can pull. Arrange enough of them around a bell and the bell can pulse, which is how a jellyfish swims. A shrug, a jet of water, a slow rise, a sink, repeat. It works, and it is beautiful, and it is weak. The contractile cells are a component of the skin, so they are limited to skin thickness and skin arrangement. You cannot build a bicep out of epithelium. The triploblastic move is to take contraction out of the epithelium entirely and give it a tissue of its own, built from mesoderm, thick, layered, organized in whatever direction the job demands, anchored to whatever needs pulling. And once muscle is its own tissue, everything opens. You can lay muscle in circles and squeeze. You can lay it in lines and shorten. You can put a muscle on one side of a joint and its antagonist on the other. You can burrow, crawl, swim fast, jump, grip, bite, chase, and flee. Consider the deep symmetry of what's happened here, because it's one of my favorite facts in biology. The same 2 proteins, actin and myosin, that a solitary eukaryotic cell used 2 billion years ago to drag a vesicle across its interior are, right now, contracting your heart. The scale changed by a factor of 100 million. The molecules did not. Evolution did not invent contraction for animals. It found contraction lying around inside a cell and built an animal around it. 5.9 The first organs. An organ is what you get when 2 or more tissue types combine into a structure that does a job none of them could do alone. By that definition, organs show up before the mesoderm does. Even a jellyfish has them, after a fashion. A tentacle is skin, which senses, plus myoepithelium, which pulls, plus nerve, which coordinates, plus a battery of cnidocytes, which fire, all working as one instrument. That is a genuine multi-tissue functional unit, and it's an impressive one. The clearest cnidarian example is the gastrovascular cavity, a single blind sac lined with endoderm, which is both the gut and the entire transport system of the animal. Food goes in, is digested, and the products diffuse from the cavity to the rest of the tissues, which are never far away. It works precisely because it doesn't have to work hard. Diffusion is fast over microscopic distances and hopeless over long ones, and a jellyfish's 2 thin cell layers are at every point, close to either the seawater outside or the cavity inside. There is nothing far from a surface, which means the gastrovascular cavity is not just a simple design. It is a cage. Any animal that relies on diffusion for transport is forbidden from ever being thick. It can be big, some jellyfish are enormous, but it must be big the way a sheet is big or a bag. Spread out, thin-walled, mostly water. To become thick, an animal needs a delivery system that doesn't depend on diffusion, a pump and pipes, and something to carry the oxygen. It needs a circulatory system. And to build one, it needs mesoderm. The proto-organs of the diploblasts are the ceiling of what two layers can achieve. The rest of this book is what happens after the ceiling comes off. 5.10 The genes that draw the map. Nothing said so far explains the most obvious thing about a body: how it knows what goes where. An embryo begins as a ball of near identical cells. Somehow it produces a head at one end and a tail at the other, always in the right order, never with the legs coming out of the neck. There must be some system that tells the cell not merely what to become, but where it is. There is, and it was found by accident, in flies, by people who were not looking for it. In the 1970s and 80s, geneticists studying Drosophila began finding mutants of a truly disturbing kind. Not flies with a defect, flies with the wrong body part in the right place. A fly with a perfectly formed leg growing out of its head where an antenna should be. A fly with a complete, functional, extra pair of wings on a segment that normally has none. The parts themselves were flawless. What had been broken was not the recipe for a leg. It was the label on a segment, the instruction telling that piece of the body which body part it was supposed to be. Change the label, and the segment obediently builds something else, perfectly, in the wrong place. The genes responsible are the Hox genes, and what they encode are switches, proteins that bind DNA and turn entire developmental programs on and off. A Hox gene does not build a leg. It says leg, and something else, downstream, does the building. Then someone looked at how they're arranged on the chromosome, and found the thing that makes this one of the most beautiful discoveries in 20th century biology. The Hox genes sit in a cluster, in a row, and the order of the genes along the chromosome is the order of the body parts they specify, from head to tail. The gene that patterns the head lies at one end of the cluster. Next to it, the gene for the segment behind the head. Next to that, the one for the next segment back, and so on in sequence, all the way to the tail. There is a map of the animal, written in order, along its own DNA, and as the embryo develops, the genes switch on in that same order, front to back, in time. So the chromosome is not only a spatial map but a schedule. Nobody expected this. There is no obvious reason genes have to be arranged this way, and yet they are in flies, and in mice, and in you. Which brings us to the fact that broke the field open. When researchers went looking for Hox genes in animals other than flies, they found them in worms, in fish, in frogs, in mice, in humans. The same genes in the same order, doing the same job, laying out a head-to-tail map, in animals whose common ancestor lived more than 500 million years ago and looked like nothing either of them. Cnidarians have a simplified version, too, patterning their single mouth to base axis. The toolkit is older than the biliterian body it now builds, and it doesn't stop with box. Consider Pax 6. In a mouse, it's a master switch for building an eye. In a fruit fly, it's a master switch for building an eye. These are eyes built on utterly different principles. The fly's is a compound eye of hundreds of separate facets. The mouse's is a single camera, with a lens and a retina. They are not the same organ. They are not built the same way. They did not evolve from a common eye. Now take the mouse's Pax 6 gene, put it into a fly, and switch it on in the fly's leg. The fly grows an eye on its leg. A fly's eye, a compound eye, correctly built by fly machinery. The mouse gene didn't bring the blueprint. It brought the command. It said "I" here, in a language the fly still understood. And the fly obeyed instructions issued by a mammal. This is the central insight of the field, called evolutionary developmental biology, Evo-Devo, and it changes how you should think about everything that follows in this book. The enormous differences between animals are, to a striking degree, not differences in the genes they have. They are differences in when, where, and how strongly the same genes are switched on. A bat's wing and a whale's flipper and your hand are the same bones, in the same order, stretched and shrunk by changing how long a few genes stay on during development. Evolution did not need to invent a new gene for the wing. It needed to change a switch, and that means novelty is cheap, in a way nobody expected, which is why what happened next could happen as fast as it did. 5.11 The explosion. For 3 billion years, the record is patient. Life invents cells. Life invents photosynthesis, and poisons the sky. Life invents the complex cell, and then sits on it for 1 billion years. Life invents the body, and makes sponges and sea fans, and quilted mats. It is unhurried. There is no rush. There has never been a rush. Then, about 540 million years ago, the rocks go mad. Within a window of roughly 20 to 25 million years, a geological blink, less than 0.5% of Earth's history, nearly every major animal body plan that exists today shows up in the fossil record. Arthropods, mollusks, echinoderms, annelids, chordates, us, in rudimentary form. Not their ancestors, not their precursors, recognizable representatives of the great phyla, arriving essentially together. It is called the Cambrian explosion, and Darwin himself regarded it as one of the gravest problems facing his theory. He said so in The Origin. He could not explain why the record began so abruptly, and admitted the case was hard to answer. He guessed the earlier record must exist and be lost. He was partly right, and the full answer took another 150 years. Why did it happen? No single cause is enough. What we have is a conspiracy of causes, each one amplifying the others. The toolkit was loaded. This is the big one, and it's why the previous section came first. Hox's genes and the rest of the regulatory kit were already in place, inherited from Precambrian ancestors. So the explosion did not require the invention of new machinery. It required the rewiring of existing machinery, and rewiring is fast. Switch a Hox gene on in a different segment, and you get a limb where there wasn't one. Evolution wasn't inventing from scratch. It was recombining with a full toolkit at speed. Oxygen rose again. The Neoproterozoics saw another surge of oxygenation in the oceans and atmosphere. And here we return to Chapter 2's bargain. Oxygen is money. Muscle is expensive. Predation is expensive. Being large and active and fast is ferociously expensive. Below some threshold of oxygen, a big active predator is simply not affordable, no matter how good your genes are. When the oxygen came, the budget arrived. Someone started eating someone. And this, I think, is the real engine. For most of life's history, nothing hunted. Animals filtered, grazed, absorbed, and drifted. The Ediacaran world of Chapter 4 has almost no evidence of predation, no bite marks, no armor, no eyes, no claws. It seems to have been, by all appearances, a peaceable place. And then something bit something. Once that happens, the world can never go back. Because predation is the one selective pressure that feeds on itself. A predator gets better, the prey must get better or die. The prey gets armor, the predator gets stronger jaws. The prey gets faster, the predator gets faster. The prey burrows down into the sediment to escape. And the sediment, which for 3 billion years had been a still, undisturbed layered mat, gets churned and aerated and turned into a whole new world to be hunted through in turn. Every improvement on one side is a catastrophe on the other, forever, with no equilibrium and no stopping point. Biologists call it the Red Queen, after the character in Through the Looking Glass who explains that here, you must run as fast as you can just to stay in the same place. That is what switched on in the Cambrian, and it has never switched off. It is running right now, in every ecosystem on Earth, and it is why a cheetah is fast and a gazelle is fast and neither one ever gets to rest. Shells appeared, independently, in lineage after lineage. Animals learn to precipitate minerals, calcium carbonate, calcium phosphate, silica into hard armor, biomineralization, which is exactly what you'd expect once predation begins. But it has a delicious side effect, and we need to be honest about it. Hard parts fossilize, and soft parts don't. Which brings us to the caveat. How much of the Cambrian explosion is real, and how much is just the moment life became visible? Some of it is preservation. The Ediacarans were there beforehand, large, complex, soft-bodied, real. The molecular clocks put the origin of animals well before the Cambrian. So the world was not empty, and then suddenly full. It was full of soft, unfossilizable things, and then those things acquired shells and started leaving a record. But not all of it is an artifact. And here's how we know. We have windows. There are a handful of places on Earth where the conditions were freakishly perfect, where fine mud buried animals so fast and so anoxically that even soft tissue was preserved. Eyes, guts, gills, legs, brains, in a few extraordinary cases. 2 of these windows are famous. The Xinjiang deposits of Yunnan, China, around 518 million years old, and the Burgess Shale, high in the Canadian Rockies, around 508 million years old, discovered in 1909 by Charles Walcott, who split a slab of shale open and found a world nobody knew had existed. Through those windows, the Cambrian is not an artifact. It is a carnival. Trilobites, and their thousands of kinds, armored and segmented, with compound eyes made of calcite, actual crystal lenses, mineral optics, the oldest good eyes on Earth. Animalocaris, a meter-long predator that swam by rippling flaps down its flanks, with a circular mouth and two great grasping appendages folded beneath enormous stalked eyes. It was the largest animal alive. It was the first monster, and the fossil hunters who first found its parts were so baffled that they classified its front limbs as a shrimp, its mouth as a jellyfish, and its body as a sea cucumber. It took 80 years to put the animal back together. Apobinia, which had 5 eyes, and a long flexible hose ending in a claw, and which it swung forward to pass food back to a mouth pointing backward underneath. When it was first presented at a scientific meeting, in 1972, the audience laughed out loud. Hallucigenia, reconstructed for years upside down and backward, walking on its spines, and waving its legs in the air, with the wrong end designated the head. The name is not an accident, and in that same water, unglamorous, and easy to overlook. Haycowichlys, a couple of centimeters long, with a notochord and a dorsal nerve cord and the beginnings of a skull. One of the first vertebrates. Everything you are was already in the water, and it was small, and nothing was afraid of it. 5.12 The template. Take the measure of what happened here. A fold in a hollow ball produced layers. 2 layers made a jellyfish. A third layer, Mesoderm, made muscle and blood and bone, and with them the possibility of an animal that could chase and be chased. A lined body cavity gave organs room to grow and a place for a circulatory system to run, which meant an animal could finally be thick. 4 tissue types, combined, became organs, and an ancient set of genetic switches, inherited from before there were bodies to switch, provided the logic that let all of it be rearranged quickly into forms that had never existed. Then oxygen rose, and something bit something, and the Red Queen started running. And in 20 million years the ocean filled with armor and eyes, and claws and legs. And here is the thing to hold onto as you turn the page. It never happened again. In the 500 million years since, evolution has produced things of extraordinary beauty and power. Flowers, forests, feathers, wings, warm blood, eyes that read, minds that wonder about their own origins. But it has not produced a new phylum. Not one. The fundamental architectures, segmented and jointed, shelled and muscular-footed, spiny and five-fold, notochord and nerve cord, were all laid down in that brief Cambrian window, and every animal since has been a variation on a theme composed in. The themes were written once. Everything after has been arrangement. You are a Cambrian body plan. Somewhere in that water, 500 million years ago, a small soft thing with a stiffening rod down its back was swimming for its life, and every choice its body had already made, the front end, the nerve cord on top, the notochord beneath it, the segmented muscles along its flanks, is a choice you are still living inside. Chapter 6 follows the others. The ones that stayed in the water and the ones that walked out of it, the arthropods and mollusks, and annelids and echinoderms, the overwhelming majority of animal life on Earth, and the strange and magnificent things they did with the same set of parts.